Okay, welcome everyone, and good evening to our program, Our Wild Neighbors, researching the flora and fauna of Prince William Forest Park. My name is Ashley Studholm. I am the Executive Director with Prince William Conservation Alliance, and this program is brought to you, we're hosting and co-hosting with several organizations that are interested in this topic, including the National Parks Conservation Association. So big shout out to Kyle Hart, Mid-County Civic Association of Prince William. So thank you, Martin Jeter and the folks over at Mid-County, and the Prince William Wildflower Society. So thank you to Nancy Veers and all the folks over at the Prince William Wildflower Society to make this event possible. So to begin, we are an environmental grassroots nonprofit focused on creating equitable and sustainable communities throughout Prince William County. And you can learn more about us and the work we do by going to pwconserve.org. And there you will find updates about all the things that we are working on. And if you go to our calendar page, there you can see we just completed an Adopt a Stream program and cleaned out over 90 pounds of trash at Cedar Run. We have our event tonight, and upcoming we will have our butterfly survey in July. And we will also have our the Kim Hosen Memorial Hike with Prince William Soil and Water and Virginia Outdoor Foundation on May 16th. So you can join us for any of all of those events, and we really appreciate um, your presence and your participation in the events that we provide. We try our best to offer these programs to the community for free and to keep it accessible to everyone. And we were able to do that even though these are not free to put together and to host because of the generous contributions of our donors. So if while you're here, you want to support us, all you have to do is click that donate button at the top here, and that will take you to um, a secure page where you can donate very easily electronically, and you can even become a recurring donor and become a monthly sustainer, which really helps us with our programming. So tonight, like I mentioned, we are diving into some of the research that is happening at Prince William Forest Park. And so Prince William Forest Park is a premier destination for residents across the region to come and delight in nature. And we're fortunate to have two national parks within our county. We have, and both of these parks provide, and all of our protected areas provide vital resources to our communities, both the physical and mental health. It provides quality habitat for wildlife and protection to our stream quality and critical waterways. So to hone in here specifically on Prince William Forest Park, for example, and it's located in the southeastern edge of the county, it protects about 15,000 acres of predominantly Piedmont forest. And this is the largest Piedmont forest in the national park system and provides a home to over 250 species of animals and over 900 species of plants. So from the black bear to the elusive freshwater sponges, which we'll be learning about today, from the towering trees to the easily overlooked but federally threatened small rural begonia, this diverse flora and fauna welcomes over 250 park visitors annually. So they really this park makes a huge contribution to our county economically as well. So whether these visitors are taking a walk to just decompress from the week, or bird watching just for the pure joy of it, or going on a bike ride and actually breathing in some fresh air while they do that. This park offers rich experiences for all of these uses. But wait, there's more. This park also provides the opportunity for scientists to study healthy ecosystems. And with habitats globally becoming increasingly fragmented due to development pressure and research opportunities are shrinking. But here in Prince William County at Prince William Forest Park, there are opportunities still, even though it is facing similar pressure. So much of this land um, around its border. So this is a map of Prince William Forest Park, and that dark green line is the legislative boundary. That is the boundary that was set by Congress. So any of these lands that is not green. So what is green is actually Prince William Forest Park. So any of these parcels that are kind of around here that are within this like darker, more fluorescent green could in the future become parkland. That is set by Congress. So the park does not have to get congressional permission to expand the boundaries within this legislative boundary. Um, and here locally, there are land use decisions that can reduce that pressure and support maybe even some of these parcels becoming future parkland, or it can continue to erode and chip away at the integrity of the park. And that is what we're seeing. There's a proposal up here to put a data center. It has been called Potomac Technology Park. It is now called Quantico Ridge. And then there's Lebenison, which is by right would have just resulted in 3 landowners that the park would need to negotiate with. And with that proposal, they might be looking at upwards to 100 to 80 landowners, which is of course much more complicated for a park to manage, especially given the atmosphere of build baby build and not necessarily getting the support from the federal government all the way down. But tonight we are focused on learning more about the really awesome research that is being done here. And we are going to start by hearing from Kristi Shelton, who is the park biologist. So Kristi, thank you so much for being here, and I'll kick it over to you.
Yeah, great. Thanks for having us, Ashley. So as Ashley introduced the park, so as the park biologist, I also serve as the park's research coordinator. So that anybody who wants to do research in our park can work through us and work with me to get that research done. Whether that is high school students working on water quality or soil health projects, or things related to things in their environmental science classes, college students or graduate students working on their projects. We have a student who's been researching the blueberry species in the park to look at hybridizations and what's happening with blueberries out in the world. We work with universities or other research partners, some of whom we're going to hear from today, whether that's through the Smithsonian Environmental Research Center or Virginia Tech and USGS to conduct research that the park is interested in as well. A speaker we weren't able to get for tonight was a recent project looking at freshwater mussels. We were working with Virginia Tech and USGS to survey the park for our mussel populations and habitat, because the Virginia Department Wildlife Resources is actually helping to breed some of these more rare mussels, and we're looking for release sites. We were chosen with Manassas and the C&O Canal to help determine if we are a place that the state can put some of these mussels that are looking for a home. On average, each year we get about 15 research permits covering all of those things, as well as some of the county research that goes on, whether for Prince William County or Fairfax County also comes to our park to collect data to compare their stream quality and fish populations to somewhere slightly less developed. But tonight, like I said, we're going to listen to 4 of our recent ongoing research projects in the park. And I think first up, we're going to hand it over to Melissa from the Smithsonian Environmental Research Center.
Thank you, Kristi. And so Melissa McCormick, she will be discussing the federally threatened small world begonia. So Melissa, and I guess just for programming, feel free to use the chat if you have any questions, but we're going to go ahead and go through each of these uh, uh, results from these fabulous researchers, and then we'll circle back and open it up for Q&A at the end. So Melissa, please take it away.
All right. Thank you so much for having me here tonight. So I'm really excited to be able to tell you a little bit about the research that I and my lab have been doing on a rare orchid, the small world Pogonia at Prince William Forest Park. And this is research that we've been doing for 18 years of monitoring and doing experiments, more recently with Kristi, but with all of her predecessors as well. And so collaboration from the park has been super important. And this is work that we've done as scientists at the Smithsonian's Environmental Research Center, but also I am currently the director of the North American Orchid Conservation Center. And so this is just an example of the type of work that we get really excited about being able to do. I want to start out by telling you a little bit about orchids that you may not know. So there are approximately 30,000 species of orchids. They make up 10% of all of the world's flowering plant species. That's a lot. They're either the most species-rich plant family or the second most, depending on whether you're an orchid fan or an aster fan. So, but they grow everywhere from the tropics to above the Arctic Circle and everywhere in between. And in the temperate regions, such as we have here, they all grow in the soil. So if you think about orchids and what you buy at Trader Joe's or Home Depot, those are almost all epiphytic orchids. They are tropical species that grow on the trees. Well, in places where it gets really cold and gets below freezing, like we do here, it's a little too cold up on the trees, and the soil provides kind of a nice buffer for them. So the orchids that we have here all grow in the soil. And Virginia has 60 native orchid species, and that's out of 232 native orchid species in the US. So you can see that about a fourth of the orchids that are native to the US occur in Virginia. And so that's really pretty impressive. However, nearly half, 48% of them, are listed as either on the watch list or rarer in the state of Virginia. And so that is an issue, it's a concern, but it's also not uncommon. So that's about the same rate as we see orchids being rare at the national and global levels as well. And part of the reasons for this have to do with these really complex life histories that orchids have. And these complex life histories make them something of a canary in the coal mine of the plant world. And what that means is that if something's going wrong with the environment or the environment is changing, the orchids are often the first species that get lost. And that's kind of a depressing way to think about it, because that means we're going to lose all of our orchids fast. But it also means that if we start losing our orchids, that there's something wrong with the environment. And so we also like to think about them as the pandas of the plant world, because you can get some really sexy, beautiful orchids. And by drawing attention to the orchids, because they have these connections to the rest of the elements of their environment that I'll tell you a little bit about next. And that by conserving the orchids, we are also preserving the rest of the environment that they need. So getting to the species of interest here. So Isotria medialoides, or small world Pogonia, is shown here in the image on the left. And can you see my cursor? Okay, so in the image on the left, you can see I've got two little plants here. It's small. It's about that big, not very big at all. And you might miss it if you were just walking casually, but it's really has a very lovely little orchid flower, a single whorl of 5 leaflets, and this happens to be two little individuals that are very close together. And it is one of the 8 United States orchid species that are federally listed as threatened or endangered. And this is a federally listed threatened species. It's not at all common. And rare plants are really hard to study. And often that's because there aren't enough of them to be able to learn what causes the plants to increase or to decrease. How do you know if they're doing better? Well, if you go from 1 to 2, yeah, that's a change, but is it just chance? Or did you actually do something right? And so learning what drives populations often takes monitoring over many, many years. And as I mentioned, we've been monitoring this species at Prince William Forest Park for 18 years now. And there's very few locations where these types of studies can be done, and even fewer that are close to population centers, and so they're relatively easy for researchers to get to. Now, it would be really easy to study Isotria if it just had a normal plant life cycle. It starts out as seeds and grows to a little plant, and then it's a flowering plant. And if it gets pollinated, then it makes more seeds. Awesome, that would be lovely. It needs light to grow when it's green. Perfect. Except to that, Isotria has a couple of other steps. So its seeds are very, very tiny. They're called dust seeds. They are very much like dust. They are microscopic. And those seeds don't have any nutrients in them to support the growing seedlings. And so for them to grow from this tiny, tiny seed into this little green vegetative plant, it takes a while. It takes a lot of effort. And it goes through this stage that we call a protocorm stage that, well, looks nothing like a plant. It's not green. It looks more like little worms or something, but it's not. It's baby orchids. And the other thing that Isotria does that makes it really hard to study is that it has this stage that we call vegetative dormancy. And what this is this is a stage where the orchid can be living just fine, growing just fine, and just not produce anything above ground for years at a time. Now, this isn't the kind of dormancy that most of our plants go through in the winter. This is an active, physiologically active, growing plant. It just didn't put anything above ground. And Isotria can do that for up to 9 years. So if you have a plant that didn't come up, is it dead? Or is it going to come back next year? You know, it's very hard to study that way. And so that is also what makes it really important to be able to do these long-term studies. Now, the other thing is, you'll note that neither the below ground vegetative dormancy stage or the protocorm stage is green. So where are they getting the energy that they need to grow? Well, the answer is fungi. And they need specific fungi. And as the work that we've done at Prince William Forest Park has allowed us to identify what fungi these orchids need. That was never known before. And so if you're trying to manage an orchid that depends very heavily on another species, in this case, a particular fungus, if you don't know what that fungus is, it's really hard to be able to effectively conserve the species. And what we found is that not only have we been able to identify that fungus, we've been able to demonstrate that that fungus is essential for germination from seeds, and it's also what is primarily supporting the orchid when it's in this below ground stage. But it also continues to contribute to the growth of the orchid in these green stages that might be dominated by light. So if we think about the fungi that Isotria needs, these fungi they're not just your run-of-the-mill fungus that lives underground. These fungi are themselves totally dependent on associating with particular trees. In this case, we've identified host trees that are beeches, oaks, hickories, and also birch trees. And those trees have to be there in order to get the fungus that Isotria needs. And what we found is that the more abundant that fungus is in the soil, the more likely that plant is to pop up and produce a leaf with the potential then to flower and produce seeds. And if there's less fungus, it's a lot more likely to hang out underground for a year. And if there's really not much of the fungus, you just don't get the orchid at all in that location. And so this means that there's a really important role for fungi, in addition to the role for light, in allowing this orchid to grow. So we've done a couple of experiments to figure out, all right, well, what can we do to help the orchid populations? And what we found, we did an experiment where we went in and we thinned the tree canopy to let in a little more light. And what we found was that we were able to increase the amount of light increase the canopy from about 6% open to about 8% open. So we're not talking clear cutting here. We're not talking anything drastic, just letting a little more light into the orchid. And what we saw was that, well, as you might expect, the green stages of the orchid, those that might be photosynthesizing, were able to increase. They were more likely to flower. They grew bigger in a year. But we didn't see the increases in plants coming out of vegetative dormancy or recruiting from seeds. And that's about what we would have expected because those stages are mostly driven by fungi. And letting in a little more light doesn't really affect the fungi. But what we found was that in one population at Prince William Forest Park, there was a tree, it was a maple tree, that died and fell over right on the edge of a colony, of a population of this orchid. And what we found was that in this case, when that tree died, that population went from only 2 individuals being up to over 80 in the course of just a few years. And so really, really impressive increases. And we also found that when that tree came down, not only did it let in more light, but that also increased the abundance of the fungi that Isotria needed. So we're getting increases from both directions. And so we said, hey, I wonder if when a tree dies, we're also seeing why are we seeing increases in the fungi? Can we make that happen? And so what we're doing right now is we're doing an experiment where we're actually killing one tree in the center of a couple of 3 different colonies, 3 different populations in 2 sites, both at Prince William Forest Park and at Fort A.P. Hill. And we have other sites as controls. But what we did was to go through and we said, all right, if removing this one tree allows this orchid to increase for quite a few years in a row, obviously we're not going to kill a bunch of trees right and left. That's not happening. But if this is something that could contribute to the management of these orchid populations on a longer time scale, then that's something that we would be interested in seeing incorporated into management plans. And so in addition to those, we have some ongoing studies, and you might have seen that I had a picture of the orchid pollinator. Well, guess what? Until last year, there were no known pollinators for Isotria. And so what we have done is we've designed these little motion detecting video cameras. And here is our little small world Pogonia orchid here. And we've been able to record pollinators on the Isotria orchids. Now, we haven't yet recorded them at Prince William Forest Park. And just before this meeting, I was consulting with Kristi as to how far along the plants are, because we are putting cameras out on Friday. And we are really hoping that we're going to get some good pollinator visits to the Isotria at Prince William Forest Park this year. So we're giving it a shot. This happens to be a site in New Jersey, and we had some from Pennsylvania as well. And then the other thing that we're doing is we're working on determining how the plants at Prince William Forest Park, how closely related they are to other Isotria within the Mid-Atlantic, and then also within the broader range for the species. Because what we want to know is there anything different about Prince William Forest Park orchids? Are they a great study organism because they happen to be representative of what we see in the Mid-Atlantic? Or conversely, are they a really unique population that we need to make sure is conserved effectively to be able to conserve the ability of the species to evolve to new conditions? And so we are incorporating all of these studies into management plans. And one of the things that's really cool is that over the years, all of the work that we've been able to do on this species is now gotten us to the point where we understand a lot about the ecology of this orchid. We learned what it needs to grow, its pollinators and mycorrhizal fungi. We learned how to manage populations that are declining. And these are populations that are on protected land. And together, this research has accomplished what is needed to support the likely delisting of this species in the next 4 years. So it's a species that was identified by the National Fish and Wildlife Federation and the U.S. Fish and Wildlife Service as a species that is likely to be able to meet the conditions needed to remove it from the threatened species list, and still allow the species to persist and continue growing. And so trying to incorporate all of this information that we've learned at Prince William is allowing us to advance our conservation of this species. So this is obviously a lot of work that has gone over 18 years with a number of different funding sources. But of course, I want to shout out to folks from my lab, National Park Service, and Kristi, as well as the Smithsonian and the North American Orchid Conservation Center. And I did mention that I have to duck out to go put some replace batteries in my pollinator cameras. But if people have questions that I could answer real quick, I will give that a shot.
Thank you so much, Melissa. And I'm sure Kristi can answer a lot of those
I'm sure she
potential
can.
questions too. So we'll move right along, but I really appreciate the work that you're doing. And I love the description of the orchids as the canary in the coal mine. But really what you're describing is like, it takes a village,
Yep.
and the integrity of the ecosystem and the relationship with the fungi is it's just so fascinating to see how everything is so interconnected and what those successional processes are that kind of leads to these ebbs and flows of the population, I think is really, really fascinating. And what is so great about Prince William Forest Park is because it's such a high quality area, that you're able to control those specific variables to answer the
Mhm.
questions that you are asking. Because these are things that are really potentially dynamic that can be altered pretty quickly if, I mean, the changes in the edge effects and things like
Yep.
that can really influence the results that you're getting. And so the results that you're gaining is really anchored in that reality. So Melissa, thank you so much. And we will include in a follow-up, if that's okay with you, for people to reach out to you if they have additional questions that Kristi potentially
Absolutely.
can't answer. But yeah, go and take care of this
I
this
just have to get
rose.
everything changed out before it gets too dark. I can't see what I'm doing.
Perfect. All right. Well, thank you, Melissa. And
Thank
we are
you.
going to move ahead and explore, something that's really overlooked, and that's characterizing Deadwood. And remember, like all soil, all the rich soil that we have comes from organic material. And so we're going to look a little bit more at characterizing Deadwood with Dasha. Maslyukova, and she will illuminate us a little bit into her research at Virginia Tech. So, Dasha, thank you so much, and I'll kick it over to you.
Thank you, Ashley. Hi, everybody. As Ashley introduced me, I am a current PhD student at the Virginia Tech Forest Resources and Environmental Conservation Department. And today I'll be telling you a little bit about my master's research that I did at Prince William Forest Park back in 2023. So to get started, let's define what downwoody material is. So in this presentation, I'll be talking about duff, litter, as well as different size classes of our woody material. So that goes all the way down from a tiny stick to big coarse woody debris, which we define as being anything over 3 inches in diameter. We also collected data on our standing and dead trees. So those are our snags at various stages of decay. But why should we care about Deadwood? Ashley has alluded to that already a little bit, as well as Melissa. And so three of the main reasons is that we need Deadwood from a wildlife perspective. Deadwood contributes to a lot of different organisms, both standing and dead, but also down and dead on the ground. We can also think of Deadwood as potential fuel that can increase the risk of wildfires or be something that you need to know the amount of if you're going to be planning a prescribed fire in the park, which Prince William Forest Park has done before as well. Deadwood is also integral to different nutrient cycling processes and returning back nutrients into our soils through the different processes. Specifically with wildlife, we have different numbers and amounts of deadwood that different organisms need. Certain studies have shown that small mammals and different amphibians, the numbers of them have been associated with having larger logs or stumps, as well as our standing and deadwood being contributors for habitat for birds and different bat species. This, I don't know if you can see my cursor, but this is actually an image from Prince William Forest Park of a previous PhD student, Dr. Samuel Fries, that did research on bats at Prince William. If we're thinking about wildlife hazard and prescribed fire, the amount of fuel that we have in the landscape can actually be a deterrent of prescribed fire because some of those larger pieces can hold on to moisture longer. And thus, if we want to have a prescribed fire for different ecological effects or to reduce the amount of fuel that we have in our forests, the deadwood can actually be a deterrent because when we're doing a prescribed fire, we might not want to be burning on the hottest day or the most days without rain. And thus, those big pieces of wood can actually break up that fuel continuity and reduce the effectiveness of our prescribed fires. However, when we're thinking about wildfires, the amount of fuels, especially with NOAA anticipating the number of days without rain, so our drought severity index in Virginia is projected to increase, as well as the precipitation amount. So thinking about the intensity of these events, the amount of fuel that we're having on the landscape can really impact the wildfire risk hazard that the park can see. And then nutrient cycling, if you look at this diagram, that dead biomass or dead organic material is at the core of three different cycles, right? Our phosphorus, carbon, our nitrogen cycles, which are very important for the way that plants are using these elements on the landscape. And as well as thinking about fungi being the thing that breaks down those materials, uses that carbon to nitrogen ratio to then do a lot of other stuff. So what do visitors currently experience at national parks? So the park has expressed concern, as well as visitors, about the amount of deadwood that they visually have seen. So here on the left, we have our Virginia pine stand with a heightened amount of deadwoody material, specifically that coarse woody debris. And on the right, we have a Liriodendron tulipiferus or tulip poplar branch that has broken off. And so this kind of represents maybe two different ideas of how deadwood accumulates on the landscape. And so also if you're a visitor and you're seeing this at the park, you might be concerned about the health of a forest if this is what you're experiencing, as well as some of the oak decline that we've seen over the last decade or more, and that we are also seeing elevated amount of quercus die off and falling down across the landscape, not just in the Piedmont region. But what I'll say is that here's me on the left looking a little bit grumpy, but still happy to be in the woods because I had to climb under and over all of that deadwood material to measure it. And so we kind of talked about the national park interests, thinking about it from a visitor safety perspective, but also from that forest health perspective. And to make sure that we know what's on the landscape and that we can be able to compare it over time. So the National Park Service provided me this kind of breakdown, which was created by NatureServe and DCR about 10 years ago now, I think. And so the dominant forest cover of the park is that mesic hardwood cover type, followed by our oak, pine, hickory, Virginia pine successional communities, and then just a little bit of floodplain and eastern hemlock areas in the park. And these are slightly aggregated, so the NatureServe and DCR have very specific definitions for the type of communities there are, but we, for this research, aggregated them a little bit more. Other thing I wanted to point out is that there have been previous dendrochronology research done at Prince William and some other parks like Rock Creek in the area, and they have identified that a lot of the trees that we're seeing, since the area was used for agriculture before, was heavily clear cut. And so this red line represents the time that the area that became Prince William Park was federally protected. And you can see that a lot of the current trees in the park were actually recruited right before or right during the time that the land became protected. So that caused that recruitment number in the species across different species. But that will come into effect a little bit later, especially as we were thinking about the succession patterns that we're seeing in the forest cover types. One other thing that I wanted to point out, and that I know this group has talked a lot about, is the increasing wildland-urban interface, and with the projected housing density to keep increasing. And these numbers or these projections are from Walsh et al. From 2015, so they might have changed a little bit. But thinking about Prince William Forest Park as being that contiguous land area, the contiguous forested area in conjunction with Quantico Marine Corps Base right under it, as being kind of this area where we do have a lot of deadwood and trees, and that's all also potentially fuel. And so when we're thinking about these projected drought indices being bigger, we can also think about the wildland urban interface as being very important when we start to think about the fire aspect of our deadwood. And so that main idea was for this project was to try and see if we can see any differences between, elevation, forest cover type, some other variables throughout the park, to really dive into what is causing these patterns of deadwood on the landscape. And we did these fixed area forest cover types, as well as different transects. So throughout our transect, we looked at different litter amounts, different duff amounts. We measured the type of coarse woody material that we found. And then we also did a full stand inventory looking at the diameter of the trees, the canopy class, and also took some brief notes on the understory composition of those species. For all of our dead and standing, so for our snags, we also looked at a snag decay scale. And the idea behind this is that this is used in some of the bat guidelines. So when the bat researchers do their stuff, they often kind of mark what kind of tree and what decay scale the bats are roosting in. And so we actually found a lot of the same types at Prince William Forest Park. So these are all images from Prince William Forest Park. So you can see that we have a variety of different snags, and all of those snags contribute to wildlife in different ways, because we know that our wildlife have different requirements for what they need. And so we use generalized linear models in our statistical analysis. And our main finding was that the percent dead basal area of a plot really was significant in also telling us the deadwood of the plot. And that kind of makes sense. If we're visually thinking about a plot that has a lot of standing and deadwood, we can assume potentially, but this research kind of proves it, that we'll also see more deadwood on the ground. And this was most pronounced with our Virginia pine cover type. So all of the Virginia pine cover type plots that we had almost double the amount of standing and deadwood than all of our hardwood plots throughout the forest. But this wasn't just true with our Virginia pine plots. We could see that relationship with our other forest cover type plots as well. But as you can see here, when we put all of them in, they weren't as strong, with our R-squared of only being a 0.3. And here we don't have to focus on the numbers too much, but you can see the percent dead basal area was very important, as well as forest cover type for a lot of our variables. And when we're looking at the total amount, so the total amount of stuff of the organic material on the landscape, we're seeing that Virginia pine cover has the highest, that has significantly higher amounts than all of our other types, with all of our hardwood sites kind of being within the same range. Now I do have to point out that our oak, pine, hickory sites also had pine in them, but there were usually more hardwoods present in that type. And some of that data is consistent with previous studies as well. Then I wanted to point out the finer fuels. So that is everything basically smaller than a log that's 3 inches in diameter. And here what's interesting is we're seeing an elevation in that floodplain area. And even though Prince William had only about 3% forest cover type in the floodplain, so dominated by sycamore, sweet gum, some elms. We are seeing also an elevated fine woody material mass there. And some of the reasoning behind that could be that the floodplain deposition events are putting that fine woody material when the river or when the streams are kind of fluctuating, is then depositing that material onto our floodplain, and then we're measuring that. And then also the legacy of emerald ash borer in our Fraxinus species throughout the park. And then the last thing I'll just point out is that litter. And litter was the only one that elevation was significant in. So thinking about how high the site was in relation to other sites. And that also kind of makes sense because we saw the pattern that as elevation increased, our litter decreased. So when we're thinking of a little mound or a topography where we're seeing a heightened area, we would maybe anticipate that litter being kind of pulled down by wind, gravity, water, erosion. And just over time, since that litter hasn't been incorporated into our soil depth layer yet, we are maybe seeing that pattern come out there. And then here we also saw, this wasn't significant, but we saw a heightened amount of oak, pine, hickory litter. And so that could be associated with higher lignin amounts in our pine and oak needles, which causes them longer to break down. And so then they're just kind of sticking around for a little bit longer. I also wanted to show you just some visual photo comparisons and to show you that there is variation between the different plots within the same forest cover type. So as you can see on the right, this is one of our more extreme sites with the Virginia pine cover type. We're seeing very high amounts of deadwood, but then we are also seeing less, some Virginia pine sites with less deadwood. But as as the data showed, it was significantly higher. And so it did really end up being either through the middle picture or that higher picture on the right is what most of that plot looked like. And so we kind of talked a little bit about succession earlier. And what we think is maybe happening here is that Virginia pine in its natural history that has been described only lives up to 100 to 150 years. And so potentially, since we're now reaching about 100 years since the establishment of the park, we are seeing the natural senescence of that species. And so this is an example of that natural senescence playing out. So now it's really important to think about what's going to be in the future of this park. What is coming up next? As you can see in a lot of these pictures, it is that American beech that is coming in, and that has its own challenges associated with it, as Prince William has been seeing elevated amounts of beech bark and beech leaf disease as well. And then here are just some, I know I'm kind of running low on time. Here are some images for our oak, pine, hickory sites. Here you can see that it was more valuable, the size of the actual material versus the individual pieces of deadwood. And so you can see that in our Quercus pine plots, it was actually basically the size of that deadwood that oftentimes influenced the amount of material in total that we would have. And then you can also see that kind of fluffier litter layer, even though in the first two pictures, it looks like it was raining when we were collecting data. And here are some images of our more hardwood sites. And so transitioning out from this, thinking about where this can be useful, these images, and now that we have associated numbers and statistics with them, can be useful in comparing other sites across Virginia, using Prince William County as an example, to understand how much deadwood a landowner might have on their property, or when we start to compare them to other national park sites as well. I quickly wanted to touch on the idea of the intermediate disturbance hypothesis that states that as we increase some disturbances, that might actually see increases in the number of biodiversity that we see. And this is maybe even what has been now kind of hinted at by Melissa in the earlier presentation too, that some of the disturbances of canopy gaps and the deadwood falling down has increased some of those orchid numbers. And so kind of thinking of Prince William Park in conjunction with our larger Piedmont forests and landscapes, and how that heterogeneity and landscape changes can influence the amount of biodiversity that we see. Yeah, I wanted to just do a quick thank you to my field crew that has helped me, as well as the National Park Service for all their great staff and funding. And I will answer questions later when we have that portion of our night. Thank you.
That's terrific, Dasha. Thank you so much. And I love that contrast between, the the role of the deadwood and the fuel load for prescribed fires versus the wildlife or the wildfires, and how it, the role of the moisture content that is there, and how that all the dynamics that play out there. So I think that's really interesting. And then looking at the actual like fuel sources, and how the Virginia pine may be, part of this like succession of this forest as it leads more, if it remains undisturbed, towards this climax forest, whatever that may be under these circumstances. So that's really cool. And these longitudinal studies that are being undertaken here can provide so much information, especially if you're able to really make sure that the conditions that you're doing this research in are steady. So I really, really appreciate that. And we will circle back. I'm sure that there's plenty of questions about your research that we'll get back to. So from here, we're going to go on to Colin Wang, who is researching the elusive freshwater sponge, which I had no idea about. And so I'm really excited to learn more about this. So Colin, please take it away.
Yeah, is my audio coming through all right?
Yeah, we hear you loud and clear.
All right, great. All right, I'll just go ahead and get started then. My name's Colin Wang. I'm an undergraduate junior at the College of William and Mary, and my presentation today is called Uncovering Cryptic Diversity and Ecology: Environmental DNA as a Valuable Methodology for Studying Freshwater Sponges. I've been working in conjunction with the National Park Service for around 2 years now, and yeah, love to share what I'm working on. So this is a small roadmap of the sections I'll be coming over, and there will be a small box in the top right to let you know what section I'm currently on. But let's back up a little at first. I think I'm in pretty good company to say that freshwater ecosystems are really neat. They are exceptionally rich in biodiversity, yet are seeing alarming rates of species extinction and decline. They only make up like 0.01% of the world's water. They're home to almost 6% of all described species and 43% of all fish species. But like I said, their rate of biodiversity decline is increasing. It's twice that of marine or terrestrial ecosystems, as this graph shows. And the valuable
Hey,
ecosystem
Colin,
services,
I'm
yes.
so sorry to interrupt you, but is there any way you can make your presentation larger? People are
Hmm.
struggling to see your presentation.
Is that any larger?
That's a lot better. Thank you so much.
All right. Yeah. Well, I guess I'll just go right back into it. One freshwater animal that maybe you've never heard of before until this is the freshwater sponge. That reaction at the start is what I get a lot of. So these freshwater sponges, they are indeed animals. They're some animals that diverged early from the Metazoa animal kingdom, but they are part of this clade called porifera. So these sponges, they can be green, they can be white, and they can even be, brown down here. And they often grow in streams on logs or rocks. So, sorry, went forward a little too quickly. But yeah, it's their inconspicuousness that I think is really interesting. Now, I love a good underdog story. I don't know if anyone else likes underdogs, but I say this to people who I present to. Freshwater sponges are quite literally the underdogs of freshwater ecology. They are key benthic feeders and water filterers of our freshwater lakes, ponds, and streams. Benthic just means that they live at the bottom of a body of water. But their water filtration ability is, I think, what makes them truly phenomenal, because one finger-sized piece of sponge can filter, more than 125 liters of water in a day. And that's harmful pollutants, toxins, and even things like microplastics removed from the water column. So this is an image that I took from Prince William Forest Park of some sponges in a stream. Maybe they've been here all this time, and maybe you've walked right past them. But even though they're so ecologically important, there's so little attention on them. And I'm going to talk about why that is. So the first reason is that they live in streams, often in murky water, under these rocks and logs that make them really hard to see. But more importantly, they're what we call cryptic species, which are species that are very difficult to identify visually or morphologically. So they sometimes have near identical appearance, but they can still have very different functional roles in the ecosystem. So you can see that actually in this previous picture, the three sponges in columns B, D, and F, they're all this one species called A. argyrosperma, but this one is white, this one is green, and the one in B is even more of a faded white. So this makes it really hard to study their ranges and where specifically they can live. Traditionally, you can sample these, or you can identify these sponges based on their morphology of their reproductive fragments that are called spicules right here. But you have to use a really fancy microscope. You would compare it to a table. So that in and of itself, it's subjective to human error, takes a lot of time. And often it's not correct because the morphology of these guys, they can change based on the water chemistry. So what you typically have is like people go out to survey sponges, they'll collect some with these reproductive fragments that are only made at certain times of the year. You know, it's not known what species make these at what times of the year. So you're not very certain about the estimates that you bring back. So that's a little graphic of what people look like typically. So that's the main reason why we still know so little about them. The key to this, and what I believe the solution to this problem is in DNA barcoding or DNA sequencing, which tells species apart based on DNA sequence and not their appearance. And I think this is especially unique and appropriate for cryptic taxa because it can, distinguish the species genomically instead of their physical appearance. So what I set out to do was use this new technique called environmental DNA. I'm sure some of you have heard of it before. It's called eDNA for short. And it's a way to track the DNA fingerprints or traces of animals or plants left behind. It's a non-invasive method that you don't have to go out and physically collect or capture or tag anything. You don't have to harm the sponges that you want to protect. All you have to do is take a water sample, bring it back to the lab, and then you can do some DNA sequencing from there. And then an important factor to keep in mind is that we know very well that eDNA can say, there's presence or absence of something here, but it's less sure if you can say that there's a quantitative estimate of species abundance. So in other words, we're sure that we can say, this sponge, bird, or fish is here or isn't here, but we're still working on being able to say how many of that species are there. And that second part is really important because being able to quantify the level of species is important across, disease control, invasive species management, and many other areas of conservation. The way you do that is through the thing called read numbers that I'll talk about, but it's really hard to control for. Now, this brings me to my research questions. So the first one, no one's used eDNA on freshwater sponges before, so can we use it to track their presence and absence? Second, are their read numbers a suitable quantitative proxy for their abundances? And then third, just because we're curious, what environmental variables are correlated with freshwater sponges? So the first step of our methods was to build an initial reference database of DNA sequences that we could then match future samples against. And then in late summer 2024, my research advisor, Dr. James Shelton, and some of our collaborators from the National Park Service, we collected 184 specimens of sponge from Prince William Forest Park using two methods. So the first method that we used was called what I'm calling physical collection, or PC. This is getting that sponge from a rock, from a log. And then the second method was using eDNA read numbers. So I'm trying to visualize that here, where we have the sponge coming from a rock, and then this is a backpack sampler, which you can use this nozzle, has a tubing that can suck up a water sample. And then you would identify the sponge based on their DNA sequence or their morphology. So that's a little bit on the methods. We collected 33 water samples using that backpack sampler. And then here's an image of me with that backpack. So just to visualize what that DNA sequence looks like a little more, this is a zoomed in close-up of the DNA sequence. You can see there are some alignment regions in green. There are some yellow variable regions as well. Now, these variable regions, that's kind of like a grocery barcode here that is going to distinguish these species apart from each other. And it's it sounds a little complicated, but it's a lot more consistent than just going off of their appearance. And then finally, here's a map of the park at which I collected my samples. We were trying to sample from both the North Fork and the South Fork of Prince William Forest Park. And these streams will then go into the Chesapeake Bay. So let me quickly mention before going into the results is that there was a previous study in 2007 that found a really interesting population of freshwater sponges. So the prairie diversity was just one sponge. And we're using eDNA now to build on that work, see how many can we find with this new method. So we're confident that after the survey, we know there are at least 8 species that are different species in the park. And let's get into some of these results. These are 2 bar graphs, one of the method 1, PC, and 2, method 2, the eDNA. And what you'll see is that there's species on the x-axis and the count on the y-axis. Now, one thing should jump out at you right away, which is that this one species, A. igyrosperma, it's really dominant in the park because it's found at high abundances from both of these methods. And our end goal here was to ask, how well do these 2 data sets match up? If the eDNA method is a suitable approach to studying sponges, it should return similar data to a traditional PC survey. So how does it do? To quantify that, we did 2 different things. First, we compared their species proportions, or how much of the sponges found at a site belong to a single species. So let's say that there's this site here. We're going to count the number of sponges using method 1, PC, and we found 5 sponges of species 1, 2 of species 2, and 3 of species 3. This is just mock data, but you can divide that by the total number of sponges to find these proportions. Now let's say we went back and we resampled that with eDNA, and we found slightly different results. There are slightly different proportions here despite being the same site. And we're trying to ask, how well do these 2 data sets match up across all sites, across all species? So that's what I'm going to show on this next scatter plot, where we are comparing the sponge proportions of PC versus eDNA on the y-axis. In a minute, I'm going to show this gray line on the 1-to-1 diagonal. And the closer that these linear regression lines fall to that line is the more that those 2 methods agree. So this looks like a lot, but essentially we have 4 different species here that both methods found. And there are 2 species here that disagreed more across the data sets, but there are 2 in the middle that showed more agreement. And you can see that they're 95% confidence interval. Okay, can you give me 5 minutes? That there's more agreement between these 2 methods. And this is important because maybe we're more cautious when quantifying these 2 species with read numbers, but we can be more confident with these 2. Here's another test that we did called a Mantell test analysis, where again, you have PC on the x-axis, eDNA on the y-axis. And again, we have this gray 1-to-1 diagonal line. And now this method is showing that there's significantly similar site assemblages between these 2 methods. And that's really important because it's a good piece of evidence in favor of eDNA for studying sponges. So this brings me to our 3rd research question, which was, what variables were correlated with sponges? We also measured the water temperature, pressure, pH, and turbidity of every site that we were at. And ultimately what we found is that turbidity and the number of sponges seem to be inversely proportional. Turbidity is like the murkiness of the water. So what we found is that like higher turbidity sites, there are fewer sponges that are found. This could be that sponges, can't live in more turbid sites, or it could be that they're just more turbid because the sponges aren't filtering the water. We unfortunately can't say just from this data alone, but that's what future studies could go into. These 2 plots at the bottom I'm showing are called NMDS ordinations, where we are grouping sites based on their turbidity. And what you can see is that there are 2 distinct clusters, red and blue, on both of these kind of graphs. So turbidity does a really good job of separating those sites by that variable. So all this is just to say that we can do a lot of cool things with freshwater sponges with eDNA. You can use it to identify them. You can use it to model their abundances. And we can use it to make hypotheses about them and actually go out to test them. And the reason all this is important, it's not just cool species, but the health of our freshwater ecosystems is directly tied to both their biodiversity and the industries they support. And we can use these new methods to, you know, study freshwater sponge diversity worldwide for scientists and conservationists. Remember, these guys are, this is important for fishing and recreation and agriculture. But yeah, these guys are filtering 125 liters of water just in a finger-sized piece of sponge. So I want to give a thank you to my research advisor, Dr. James Shelton, our NPS coordinator, Kristi Shelton, our Willie Mary Lab, and the members of the National Park Service. We had a great time sampling in the parks. The sites were really accessible and the staff were very helpful and friendly. So I want to thank you all for your funding and support.
Wow, that is so fantastic. Thank you so much, Colin. I mean, the fact that you're able to build on previous research and then not just dive into these discoveries, but really test different methods to determine what the truth is on the ground. Is really, really fascinating and refining those methods to see, what is the best approach here, and that kind of iterative process of, what is that ground truthing with, the methods to use to discover that and then verifying that, that's really, really, really fascinating. And that relationship to really tease apart between the diversity and the presence of the sponges and turbidity. And I think that that will be really important moving forward, especially as there's additional pressure at the headwaters of Quantico Creek. And I really hope that, our um, our decision makers decide to really maintain that integrity of the park so that you can really tease that out appropriately. Um, so that's really great. And there are some questions in the chat for you, so feel free to circle back and start answering those, but we'll read those answers out loud so everyone has the benefit of learning from that process. And we're just going to jump right in to Elizabeth Hunter from Virginia Tech, who will take us through turtles. And I think we're focusing on the box turtle, if I'm not mistaken, which is definitely one of our charismatic, perhaps not megafauna, but certainly charismatic fauna. So Elizabeth, thank you so much for being here, and I'll let you share your screen and we can jump into the research that you've been doing.
Okay, great. Well, thank you so much for having me. And yeah, this is part of a larger project on three at-risk turtle species: wood turtle, spotted turtle, and eastern box turtle. Um, and all of these are called at-risk species. They are not listed federally, but wood turtle is being considered for listing, and all of them have different statuses within the states of their ranges, um, but all of conservation concern. We will be focusing on the box turtle today for Prince William, though. Just before we dive in, I just wanted to introduce the turtle team. So I'm Elizabeth Hunter, and I and Mark Ford are with the U.S. Geological Survey, Virginia Cooperative Fish and Wildlife Research Unit that is housed at Virginia Tech. And then our crew that has collected the field data and will continue to collect more field data includes Max Jones, Quinn Bozek, Zoe McMillan, and Alondra Medina. And then Kevin Loop is doing genetics for us. So as I mentioned, this is part of a larger project that we are doing at 15 national parks across the mid-Atlantic, kind of writ large. And the objectives for this project include to estimate the occupancy, relative abundance, and habitat use for these three turtle species, identify climate refugia for thinking about where can these species persist with climate change, and then determining the genetic structuring within and among these parks. And all three of these objectives contribute to identifying potential release locations. And I'm going to get into a little bit more background about why that is of interest. And this is of interest because turtles are illegally collected for the wildlife trade. They have many different uses. They're used for pets, they're used for traditional medicine. They're shipped all over the world, collected from here in the US and shipped all over the world. A recent paper, Easter et al. 2023, examined cases in the media from the last 20 or so years, and there were 24,000 turtles in just those 54 cases. Box and spotted turtles are traded the most, and those are two of our focal species. And then I and several folks in my team are part of this collaborative to combat the illegal trade in turtles, CCITT. And folks in that group have looked at additional cases that weren't in that Easter paper, and there have been at least 140,000 confiscated turtles in the last 10 years. And this is just really the tip of the iceberg. This is a huge problem, for turtles that are already facing lots of threats like habitat loss, climate change, etc. So, we are trying to tackle this problem with enforcement, and so turtles get confiscated at ports, like airports and things like that, and they are confiscated, and then hopefully, if they're healthy enough, they can be repatriated to the wild. But the question is where. So what we need to know is population assessments for wild populations to see if they could support additional turtles. We need disease profiles. We don't want to be moving diseases into wild populations. And then we need to understand the genetic landscape. So the question is, can national park lands be suitable recipient sites for reintroduction of these confiscated turtles? So for part of our project, we're focusing on eastern box turtles. This is work that we've been doing at Rock Creek, Prince William, the National Capital Parks East, NACE, and then the George Washington Memorial Parkway parks as well. And as I mentioned earlier, eastern box turtles are the most heavily collected and confiscated turtle in the US by far. As you know, as was mentioned, they are much loved to their detriment, sometimes species, and so people want them. So they're so heavily collected and confiscated, there's this need for reintroduction locations. But disease and genetic fit are a concern. So what I'll talk to you about today is the disease component. So we have been sampling box turtles at these parks, and we've been testing for all of these pathogens listed here. We partner with the University of Illinois, Matt Allender, who's really the expert in turtle diseases, using PCR screening. And we're already hearing a bit about DNA. I'm not going to get into it more than that. So really big concern diseases are things like ranavirus, as those can cause big die-offs, but all these other pathogens can lead to health concerns that are problematic at a population level. While we were doing field work, we also noted that many turtles in these parks had shell lesions. So you can hopefully see within those red circled areas that there are kind of some pitting or the shell's almost eaten away. And we, after observing many turtles with these lesions, we wanted to know, what's the deal with these lesions? So our objectives were to assess the presence and drivers of these shell lesions across a gradient of anthropogenic development. And the parks that we were sampling have that nice gradient, with Prince William being the least developed. And then we wanted to explore the potential pathogen contributions to these shell lesions. Are they caused by, our main target was a fungal pathogen called Amidomyces that has really spiked in prevalence for some aquatic species and caused some really big concerns. So we sampled 269 turtles across these 3 or 4 parks. We had the park that had the most was Prince William. It's a really good park for finding box turtles. Out of the, we only were able to test a subset of those. So 58 individual swabs were sent for testing, and the majority of those had shell lesions. So the pathogens that we detected included herpes virus, that was at Prince William. This is a very common pathogen in turtles generally, and in box turtles specifically. It's likely host adapted, so it's really no concern. It's actually good that we're detecting it. So, it's so common that you would be concerned if you weren't detecting it at all. Herpes virus 2, which was also at Prince William and at Rock Creek. It's also common, but it's not host adapted. It can lead to cancer in turtles. Adenovirus, which we just found at Rock Creek, it's very prevalent in the environment. It can be linked with mortality, but it's really only in very stressed animals that it is linked with mortality. So it's not a problem in the wild, but it would be a problem for those confiscated turtles. And then chlamydia, which we only found in Rock Creek. This is an emerging disease in turtles. Not much is really known right now, so it's a good thing to keep your eye on to make sure that we don't have, large die-offs caused by it. We didn't see any mitomyces, and this was our suspected pathogen for those lesions. So that was good. And we also saw that lesions were not more common in turtles testing positive for pathogens. So there wasn't really a pathogen link for the lesions. What we did see for explaining the lesions, so contrary, our expectation was that the lesions might be more common closer to water, as we thought water might be a pathogen carrier, and we have seen in the literature that aquatic turtles are having big impacts from mitomyces and these shell lesions. But this figure on the left is showing us that the predicted probability of a lesion actually goes down as there's more water in the location of where we found the turtle. So it's not a waterborne pathogen issue. And then the second graph is showing us that the probability of a lesion goes up with larger turtle size. So lesions were more common in larger turtles. And then we didn't see any really clear patterns across parks in different regions. We had thought maybe we'd see more lesions or more pathogen prevalence in more developed parks. And it's not really, the parks that were in the DC area, more developed areas, there's not really a clear pattern here with on this side is more probability of having a lesion and this side is less. And we're not seeing a clear pattern there with development. So what does this mean for management? So the lesions in the parks we sampled were not clearly related to pathogens, but they were more common in larger turtles. So they may just be acquired as turtles age, and maybe they're not detrimental to survival. And maybe we're just noticing them more because we're becoming more concerned about turtle health with this whole illegal trade issue. We saw no major pathogen concerns, for instance, no ranavirus, which is the really concerning one. But it is interesting that the pathogens that were the least benign were all in Rock Creek, which is the most urban park. Just some final thoughts. When considering disease risk for these turtle releases, it is important that the thinking now is it's important to match the disease profile of the wild population and any released turtles. So if we had confiscated turtles that had, this adenovirus, herpes virus 2, then we would at least know, okay, the turtles in these parks already have those. So it's not a concern that we would be introducing them. So it's a good thing to continue to develop these lists of pathogens at potential release sites. And actually, an idea now is that we could maybe get these lists of pathogens through eDNA, like you just heard about, as opposed to sampling the turtles themselves. And that would be a faster and more cost-effective way to get those lists. So with that, I'd like to thank all the park staff for supporting this project. We are funded by the National Park Foundation, and want to thank all of our field technicians and the National Park Service for giving us access to these great parks. And if there's time for questions, I think maybe we're at the question part.
Absolutely. Thank you so much, Elizabeth. This was fascinating, and it's so important. It really highlights, I think, attention to, these diseases have really started, like it's, I don't know, it doesn't seem like it's been going on for that long. And so it's really important to have, the Prince William Forest Park was categorized as the least developed. And it's really important to kind of maintain that those variables so you can see how these different factors influence things. And it was really fascinating to learn that, okay, maybe there's some like baseline of some of these pathogens that are proliferating throughout the population. And then you can build on that. Other researchers can build on that the findings that you discovered. So really, really great. Thank you, Elizabeth. So we are going to move into the Q&A, and to all of our researchers, as you see questions pop into the chat, feel free to answer them. We will be sharing the chat with all of the participants that are here with us tonight, but we will open it up now for a conversation and Q&A to interact with the researchers that have done just such terrific work here, and we have this opportunity to pick their brains a little bit. So I'm going to start us off with Elizabeth Morris Hughes. If you are still here, if you would like to unmute and come in, I see that you have a couple of questions. And why don't you come in and start with your first one, and we'll go from there. Elizabeth? Let's see if I can ask her to unmute. All right, Elizabeth, we'll circle back to you. You can raise your hand. Okay, there you are. Great.
Yeah, I'm
Elizabeth.
sorry. I wrote in my questions. Can you see them on the
I
chat?
can. I just wanted to see if you wanted to come in and ask them, since it didn't look
Well,
like
the
they
one I
had.
didn't get to write down is the last speaker's, a question for the last speaker,
Okay, go for it.
which was I understood that the research that she discussed was triggered by lesions on the turtles. But were these lesions on turtles already resident in the park, or was the concern because there were lesions on the turtles that were being captured and wanted to be released? Because the results of the research were they came up with very good disease patterns for the different park areas. And that at least gave them a background so that when they had turtles that they had rescued and that they needed to release back into the parks, they needn't be concerned if they knew the disease patterns in the parks that they were going to release them into. But my question was, what was the original, which were the turtles that had the lesions? Were they on all the turtles, both the ones that were about to be rescued and then released, or were they already in resident park areas?
Yeah, so good question. So the turtles that we were sampling were all wild turtles in the park, so in
Okay.
Prince William and Rock Creek. But these lesions have been noticed in these confiscated turtles, and folks in the community have not been sure whether this is of concern. Is this is this something that we should think about when we want to release turtles? Is this an indication that we shouldn't be releasing turtles? And so our study is showing that in these wild populations, the lesions are likely benign. They're not an indication of any, co-infection with something that's really of concern. And because the larger turtles have these lesions, that means as turtles age, they get bigger. And so if you are a big turtle and you have lesions, that means you've survived even though you have lesions. So it's not necessarily a problem for survival. Does that answer your question?
Yes, thank you. Yeah.
Mhm.
Thanks, Elizabeth. Kristi, there's a question here. If you could discuss a little bit more about the potential pollinators for the small world begonia
Mm,
orchid.
that was one of my questions. Yeah.
Yeah, but it hasn't been answered, so we might as well
No, no,
jump
absolutely.
into it.
Thank you. Thank you.
Thank you, Elizabeth.
Yeah, so Melissa mentioned it a little bit, that has been a big unknown for a long time for this plant.
Yeah.
For the most part, it was assumed to have been self-pollinated or wind-pollinated. Because there were just no one was catching any sort of pollinators visiting these flowers. They're so small, they're hard to see. But the Melissa's success last year, they were able to photograph two different kinds of bees, of very small bees,
Okay.
and a fly that was visiting these plants during the kind of most active pollinator season. Sorry, my cat's tail just popped right up in my face. So I don't know for sure if we've gotten all the way down to the individual species they were able to tell it's two different kinds of bees. But yeah, so it was a success last year, and hopefully we'll be getting our cameras out in our park and we can see who's visiting our flowers in our region.
Yeah, that's amazing. I went to the orchid exhibition in the African American History Museum. There's been an orchid exhibition there. And I was aware there was a problem with pollinating orchids, but my goodness. So if they've even found, at least they've got the insects anyhow that are potentials. Yeah.
Mhm.
That's great. Yeah. Thank you.
And Elizabeth, while you're here, your question to Dasha hasn't been answered yet either about the dendrochronology, and if you could, so Dasha, could you speak a little bit more about how you see the ultimate research results being applied in park or private land management plans?
Yeah, for sure. One of the interesting things about Deadwood research is that we don't have a lot of data for this Mid-Atlantic and Virginia area. A lot of the studies have either been in the Southern Appalachians, out west, or in Pennsylvania, kind of northern, northeast region. And so even having the data, and now that we've expanded to other sites like Rock Creek, Catoctin Mountain Park, we also last summer went down to Crow's Nest in Stafford County. And so now we have this gradient, right, from coastal plain to the mountains of Catoctin, and with Prince William being in that Piedmont coastal plain area, to even have a baseline
Yeah.
for
Yeah.
the Deadwood research. However, I really think that with the creating the photo guide, that's something that can be used by both parks and private land managers, especially if we succeed in pairing it with existing research that's out there for Deadwood. So, for example, if somebody wants to increase their like a specific species, potentially being able to compare what they can see in the picture with specific Deadwood kind of guidelines from other existing research about some of the animals that people might want to see, could be something that is interesting to people. And then kind of the last reason why is thinking of it from that fuels perspective. So
Yeah.
you can estimate by using the data that we collected, we can put it into systems that the Forest Service runs, such as Behave Plus, which can, or Virginia Smoke, which can estimate conditions for prescribed fire or wildfire. And so then managers can also use this visual assessment paired with our statistically sound numbers to basically then predict certain conditions if they're planning a prescribed fire burn, or if they want to predict potential wildfire spread. So that's kind of the implications of the Deadwood research.
That gives more control. Yeah. Okay. Thank you.
Terrific, thank you. So Colin's been busy answering some of those questions in the chat, so I invite you all there, and we'll certainly share those answers when we follow up. But Parker, you had a question for Elizabeth Hunter and the research on turtles. Do you want to come in and ask about how you would keep an eye on turtle chlamydia?
Oh yeah, I just it's really interesting to hear these different diseases in the frame of turtles. One thing I thought is like herpes putting the herp into herpes there. But she mostly answered that. She's like, keep an eye on this. I'm like, well, how would you even do that? But she was talking about like researchers basically.
Yeah. Yeah, it's not something that your casual herper could do. You need to do the DNA analysis. But in terms of being worried about diseases for jumping to humans, it's a different herpes virus than human
Yeah,
herpes
yeah.
virus. But salmonella is the one that can go between, and all turtles carry salmonella, and so it is a concern for touching turtles. So if you do touch a turtle, use a hand sanitizer.
Well, it's good to know because I feel like it's really common for people to see a turtle and pick it up.
Yes, yeah. Generally, we say you don't, enjoy looking at it. You don't need to touch it for the disease concern and for disturbance. Obviously, if it's crossing the road and you want to help it cross the road, then that is much appreciated as road mortality is a big concern for turtles. Help it go the way that it was going, unless it's going to be in danger on the other side.
It's such a good reminder, and it's definitely something that I do often, especially during certain times of the year. It seems like, you're driving to one of these areas. I go to Meadowood a lot. That's based in Fairfax, and going down that road, seeing turtles and having to stop and let them get them to the side that they're pointing towards, that they're wanting to go towards, because often if you put them on the opposite side, they're just going to turn around and try again. So, anyway,
Yep, this time of year, it's females looking for nest sites, so they know where they want to go.
absolutely, absolutely. Um, and that seems to be a universal delight for a lot of people. I did that once, and there was like a service worker, like electrician, and I did it, and he like stopped and yelled out his window, "Save the turtle!" And it
Right.
just, it was
Nice.
so fun. Um, are there any more questions here? Feel free to unmute and come on in and
But
ask the
excuse me,
last question.
I have my last one, which was for Mr. Wang.
Okay.
He's very interested about the eDNA sampling, the water sampling. This may sound crazy, but what I want to know was, does he take the samples from exactly the habitat where where the sponges are, or is it just from the general water habitat? In other words, are you getting indirect samples of the presence of the sponges, or are you getting direct samples of the presence of the sponges?
Yeah, I think Colin had to hop off, but I can talk a little bit to that. So when we were staging out our more manual surveys as well as those eDNA surveys, we basically selected sections of the creek that seemed like they'd be good habitat in terms of like depths and
Mm.
trying to be as successful as we can and trying to find what species are here, versus a little bit more of how you might randomly survey for other things
All
to
right.
determine distribution and things like that. So each of the samples were collected in places that we knew we had visually seen the sponges at. And then there is a whole process of like cleaning out the machine and the filters, and how those samples are collected to make sure that there isn't like direct cross contamination between the sites. And the sites are pretty far apart, and several of these were separated out by either like a geological or man-made feature like a dam or a lake. So we had kind of barriers separating out to try to minimize like the population upstream influencing all of the data downstream. So it is a little bit more site specific
Yeah.
because of that reason. And then one thing I did want to point out, if you guys can remember when he quickly showed up those two graphs with the different colors, like the red clusters and the blue clusters next to each other. It was really interesting. So the red was I think the North Fork of the Quantico Creek, and the blue was the South Fork of the Quantico Creek. So both forks of the creek, even though they eventually meet up and become one creek, had very different species distributions of those different species, which we thought was really, really interesting. So hopefully that helped answer that question.
Yes, thank you. Thank you very much.
Thanks, Kristi. And we'll go one more. So Diana Harris, if you want to unmute, come on in.
Okay, if I can figure out how to do that.
Oh, we hear you just fine. So go ahead.
So the question was going to be for the young man that was with the sponges, but I'm sure it's kind of like it will cover a lot. This question that I've had for a long time. You know how you have wetland plants like cattails and things, and people will actually make these little wetland areas so that they will filter out heavy metals and toxins and different pollutants in that. And it sounds so great, but I mean, obviously metals are an element, and when these things die, don't they go right back into the water again and everything, and just like the sponges? I mean, it's great that they filter it, but I'm like, but when the sponge dies, then it all goes back in the water again, right? Isn't that so, or am I missing something? I've wondered about it for a couple of years, actually.
Yeah, so it's a little bit about you're right. Yes, once it dies, it goes back into the system, but it's kind of that storage piece. So if we think of things like carbon buildup from pollution and things like that, and then we have trees that store all of that carbon through filtering the air. So the same thing's kind of in the water, right? So all of these things kind of get stored, and then they are like released more slowly, basically. So yeah, like you said, once it kind of accumulates and it's filtered by these organisms, it's stored there for a while, and so it's kind of out of the general environment, and it kind of is released a little bit more slowly over time as they then die and break down and decay.
Which is why it's good to, I don't mean to interrupt, but that's why it's good to keep those things alive, right?
Exactly, yes.
So, which is, I guess that's the hard thing to do. Yeah. Okay. I just wondered about that. Everybody celebrating because things, filter, and I'm like, yeah, but how about when they die? But yeah, you're right. At least for a while, and then the slow release would be better. And as long as we can keep these organisms alive, they can keep, doing that filtering and stuff. Okay,
I don't
thanks.
know for sponges, but for wetland plants, they will accumulate sediment around them, and then when they die, they become part of the soil. Every wetland is very impermanent. It's not going to be a wetland for long. It's going to turn into an upland eventually, and a freshwater wetland. And so it's accumulating in the soil. And so if you don't disturb that soil again, then it is locked in there. So I don't know how it works in stream systems, but in wetlands, that is a permanent storage.
Uh-huh.
Until it's disturbed.
Yes,
Yeah,
until we dig
right.
it up.
Until it's disturbed. Okay. Yeah. Okay.
That's a really great question, Diana. Thank you so much.
It's been a great show. I don't know what you'd call it, but I really enjoyed listening to it all. It's really been nice.
Yeah.
Thank you.
So please help me, everyone, to thank our researchers and the terrific work that they are doing to help illuminate these natural mysteries that can only really be discovered in places like Prince William Forest Park. So to all of you, to Dasha, to Elizabeth, to Melissa and Colin, who had to hop off, I just want to thank you so much for sharing the knowledge that you've been generating. You've been generating knowledge and for the opportunity to share that with us in the community. We really, really appreciate it. And and again, everyone that is on the call, we will provide the chat and we'll upload this presentation to our YouTube channel. And remember, this research is possible because of the health of Prince William Forest Park. And when these proposals go in, you do have an opportunity to speak during public comment time or during public hearings to voice your concerns. So I do encourage you to pay attention and learn about some of those proposals that are going in and speak to them, so that this research can really continue to flourish. So again, and Kristi, thank you so much to you as well in working with me to highlight the terrific research that's been going on. And have a terrific evening, everyone, and thank you so much.
Thank you.
Thank you.
All right. Have a good night.
Thank you.
You
Good
too.
night. Thanks, everyone.
Thank you, Diana. That was a really great question.
Oh, thanks. Thank you.
Yeah. Yeah. Mhm. And Diane, yeah, the MidCo meetings, the monthly MidCo meetings are really helpful to get updates on development proposals. So Diane, thank you so much for highlighting that and for joining the discussion. All right, everyone. Well, I will go ahead and end, and thank you again, and to be continued. Take care. Thanks,
Thank you,
Kristi.
guys. Yes, thanks. Thanks, everyone, for coming. Thank you,
Thank
Dasha.
you.
All
See you
right, have a
later.
good